episode of Anthropology lecture series. Today we will see what gene expression is. This was a question asked in UPSC Civil Services Mains 2023 examination. So what is gene expression? So gene expression is a process by which the information that is stored in our genes is used to make some functional end products, mostly proteins. This is in fact the bridge between genotype and phenotype. So here, in simple terms, it is where DNA produces a protein. And there are two stages in this. First is transcription and the next is translation. In transcription process, DNA produces an mRNA and an RNA strand, which is a messenger RNA. And this messenger RNA produces a protein from amino acids. So if we see the whole process, the entire process happens inside a cell. As we know, DNA is located inside the nucleus. This DNA becomes a single-stranded messenger RNA. The entire process will be shown in the next slide. So this is just the overall view. And this mRNA, it modifies and comes out of the nucleus and goes to the cytoplasm. And here we have the ribosome. Ribosome is the protein factories of our cell. So as mRNA binds with ribosome, it produces proteins. So this is the entire process. Now looking into the details of this process, this is the first process, which is called transcription. From a DNA, an RNA strand is produced. So first, there is unwinding of the DNA. Here, there is a region called the promoter region. And in this region are RNA polymerase, which is also a protein. This protein binds to the promoter region and an mRNA strand is formed. And so here this is a process. This is our DNA molecule. There is a gene here, and from this gene we get a DNA strand, and from this DNA strand through transcription we get a mRNA strand. And see, this is a complementary base pairs. For A we have U, for C we have G, like that. So here we can find triplets. These triplets are called codons. So each codon will give a particular amino acid. That is what happens in the translation process from mRNA to protein. So this is what happens here in the translation process. mRNA is turned into protein. Once again, moving to this slide, as mRNA moves out of this nucleus, there are some modifications that happens in this mRNA. That is, there are two parts in an mRNA called the introns and exons. Introns are the parts where, I'll write it here, introns are the non-coding part, whereas exons are the coding part. For our translation process, what we need is only the coding part, the exons. So the non-coding intron parts are spliced off. After the process of splicing, mRNA goes and binds with the RNA polymerase. Here, this mRNA is identified by a transfer RNA. And this transfer RNA has specific amino acid linked with it. And this linkage gives a protein sequence. And that is our end result. So that's the end process of our translation. The final product is a protein. So that's the process of translation. So this process is highly regulated in our cellular level. This is because we should ensure our cells produce the right amount of protein at the right time, and the right protein is produced. That is why this entire process is regulated. Now, what is the importance or significance of gene expression? So now let's see the significance of gene expression. Gene expression is highly important from growth, development, and even response to stimuli is based on gene expression. And the other fields where gene expression can be useful is in medicine to develop treatments like gene therapy, and to identify what kind of diseases can be treated with gene therapy can be studied with gene expression. Then the next important application is in evolutionary biology. By studying the evolution of gene expression, we can study human evolution. The next important place is biotechnology, where gene expression is used. In biotechnology, we can engineer genes engineer various organisms, and produce new organisms through concepts used in gene expression. So that's all for today's lecture. We'll meet again in another episode. Thank you.